So, welcome everybody and welcome everyone back to the first IIT seminar of this term. And I am very excited to have Rob Salguero-Gomez here from Oxford University. And he is going to be talking to us about new method to classify various life history adaptations. So without further ado, please, Rob, the floor is yours.
Thank you so much, Yuri. Thank you to the organizers of this great initiative, the Theoretical Ecology webinar, and thank you all for tuning in, regardless of what time zone you're currently based in. So I think that I don't probably need to spend too much time on this slide. Ecology needs better prediction. There's been many calls being made in terms of theoretical frameworks and calls for the synergies between theories and data to be able to do more accurate forecasting. And in order to forecast, we need to first understand. The plea that I'm going to put to this audience today is that perhaps ecology, as a relatively young science, can take a page from the book of our older sister disciplines, such as mathematics, physics, and chemistry. Those three disciplines have got a great deal of prediction. And an example that I want to present to you today is the elemental table, the chemical table of the elements, which has got over 300 years in existence. And what it does is it creates a really homogeneous way of classifying elements and two very simple principles. The first one is how many electrons are there in the outermost layer. And the second one is how many layers of electrons within the atom. Based on these two very simple axes of variation through time, researchers have been able to classify species and to even predict that between molybdenum and ruthenium, there should be a new species that will pop up in a matter of decades, that being technetium. It's the simplicity of this way of organizing elements and the predictive nature of this table that has revolutionized inorganic chemistry. And it's based on this simplicity, on this elegance that I want to present to you some work today that makes the case that perhaps we can also think about life history strategies. There you go, technetium just popped up in the table as a table of three-dimensional arrays of different demographic properties. So I've mentioned that I study life history traits, and what I'm going to be presenting to you is a way to classify them. A lot of the research that we do in my group at the biology department at Oxford has to do with understanding how species go about making a living, and we use demographic methods and demographic data. From this demographic data, we can derive life history traits and start to explore what are the limits of what's possible and not possible. Here you have got a really nice array from within the animal kingdom on the top row and the plant kingdom at the bottom row of species that have got extreme life history traits and extreme moments of their demography. The first column, species that take significant amounts of breaks, like some rodents and orchids. The second column, species that take a long migratory pathway or dispersal pathway. On the third column, species of animals and plants that invest a lot into reproduction. In the case of corals and orchids, some are known to produce 3 million propagules in every reproductive bout. Species that have the current Guinness World Record of growth, such as the blue whale, which in its adult stage is able to put on 80 kilos every day. Let that just sit down for a second. Picture what that means, 80 kilos every day. So that's the weight of a normal human and adults every day. And also bamboos, which for those of you who might think of plants as inanimate creatures, allow me to correct you, bamboos can grow up to 90 centimeters, in some cases every day as well, which is quite remarkable. Last but not least, extremes of longevity, which is an area of research that has attracted quite a lot of attention, and it's no an exception in my group. The beautiful, highly charismatic beauty at the top right is the Greenland Shark. Was estimated to have a maximum longevity of 450 years. It was featured in Science 2 years ago. And the bottom creature is the bruscon pine, which for which there are some specimens that have reached over 5,000 years. So to contextualize, what does 5,000 years mean? What it means is that the Egyptian empire was in early stages of development when some of these individuals of that species were already juveniles. So understanding how come that there's such high degree of variation has attracted the attention of a lot of ecologists and evolutionary biologists. One way to organize the variation is through the term life history trait. A life history trait is an event that describes a key transition along the lifespan of an organism. So when you think in humans, for instance, when we're born will be a life history trait. When you first go to school, in some countries, when you're formally introduced to society, Mexico, for instance, in the Fiesta de Quinceañera, when you go on to graduate from your studies and move on to your highly paid job as a pizza delivery, when you, if you decide so, marry, couple up, mix up, however you want to call it, how many kids you have, how much time in between reproductive events, how many reproductive events. Retirement age is also a very important life history trait because it determines a lot of the economics of modern societies. And back in the 1700s and the 1800s, the ultimate transition was quite a remarkable one: mortality. Every single one of these life history traits can be thought of a photograph in your album, and the album that perhaps nowadays people don't really follow because we have got things like smartphone technology, and now we take photos for everything. But back in the day, when you actually had to spend the time and the money to deliver the photographs, you'll be a bit more moderate, perhaps, in terms of when you decided to take a photo. So the photos that I present to you today are the key photos that represent a standard life history of a human. Now think of the different life history traits that define the life cycles and therefore the demographies of different species around the globe. And the first picture that can come to mind is that of a messy field where each species is represented by a different currency. You have got Australian dollars, you have got yens, you have got pounds, you have got euros. But in reality, if you think about it, common to all of these species, from lions to dandelions, from humans all the way to bacteria, is the three key vital rates that sustain those life history traits, and those are reproduction, development, and survival. So once we have got good information about survival, development, and reproduction, we can really start to launch global analysis that help us understand what are the ways in which these species go about making a living. Can we classify them? And can we actually classify them in a way that helps us predict emerging properties from those species populations and the environment, the environments in which they have evolved? So that idea may sound quite appealing. And so for quite a few decades, researchers have gone out to the field and they've been following the rates of survival, of growth, and of reproduction of mostly charismatic animals. A lot of mammal demography work has been done. A lot of bird work has been done as well. But what I've taken upon myself and in my research group is to try to expand what we know about the demographies across the tree of life. I myself have a background in plant ecology, but I now work a lot also with animals. And my research goal is to try to understand the mechanisms that shape the demographies regardless of taxonomic affiliation. Just to make you aware of what we started in that beautiful video from Wanson, that little corner, that's where most of the demographic knowledge that we currently have comes from mammals. So what I have done to preach by example, I suppose, is to acquire, digitize, error check, complement, and make fully open access heaps and heaps of demographic data. These demographic data sets are called compadre and comadre, and you're going to be hearing a lot of them today. If you don't speak this beautiful language, that it is Spanish, those two words might sound quite similar. So just to differentiate, and if the word contains a P, I'm talking about plants. So compadre is a database of demographic information in the shape of matrix population models for plants. And if I don't say P, comadre, then I'm talking about a sister database of demographic information in the shape of matrix population models for animals. And hopefully, Yuri, please correct me if this didn't work. You can also see our websites. Can you see that, Yuri?
Yes, we can see that. Thanks.
Fantastic. I just did that really impromptu transition. So this website is the open access database upon which I'm going to be presenting some of the work. And if you zoom in, you can see where every single natural population is coming from. All of these natural populations contain demographic information through time. Green is a plant, blue is an animal, okay? We've got, as you might predict, we have got a fair amount of information from North America and from Europe. And I'll be telling you a bit more about some of the current challenges that we have in some of the macroecological analysis, one of which is the biases and where these data are coming from. Right. So in addition to these two databases, we recently launched a third database called Padrino. This is work led by Sam Levin, a PhD student that I co-advised with Tiffany Knights at IDIF. And together, these three databases, so Padrino contains demographic information in the shape of an IPM, an integral projection model. The other two contain information in the shape of matrix population models. So common to these two models is that they are time discrete models. So individuals go to the field and they track changes in survival, growth, and reproduction from one discrete time to the next. They're not continuous time models. The difference between both of them is that in a matrix population model, the life cycle of a species is discretized, and in an IPM is not. It's we tend to think of life cycles in terms of more continuous pathways. The information comes from the field. And here you can see a pre-senescent Rob back in my PhD collecting demographic information in Utah. Data get digitized, models get parameterized, and then they get published. And then you've got a team of students that I co-lead together with other institutions, digitizing, error checking, complementing, and making all of that information open access. This information, these demographic approaches have become really popular in population ecology. And part of the reason why that is that they're relatively amenable to the parameterization with a relatively low degree of expertise. But in addition to how relatively easy they are to work with, they also provide a really long list of demographic properties that one can derive, both in terms of population level properties like population growth rate, rates of resilience, sensitivities, etc., but also those live history traits that I was just talking about. So we can derive from a matrix population model or from an integral projection model, mean life expectancy, rates of senescence, age and maturity. So every single photograph along the life cycle that will define the key transitions in any species for which we have got data. And this data, just to show you, have been growing exponentially through time. On the left, bottom left, you have got a cumulative increase of matrix population models in this case for the plant kingdom, and the same is depicted on the bottom right for the animal kingdom. This is another way of representing the map that I showed you before. So dark green corresponds to matrix population models from the plant kingdom, blue, matrix population models from the animal kingdom, and light green, integral projection models, regardless of taxonomic affiliation. So Padrino includes both plants and animals. So what this means is that we have got a unique opportunity to go back to basics. We have got a unique opportunity to go test ecological theory with high resolution demographic information that are more or less globally distributed. And I do want to emphasize more or less, because we would love to fill in the biogeographic regions around the globe where either little or no demographic information exists. And those are pretty obvious to me, at least. There's certain countries in South Africa, in South America, Africa, Asia, for which we have got virtually no knowledge. And together with the taxonomic, with the biogeographic biases, we also know that we have got some important taxonomic biases. When it comes to demography, a lot of the demographic information in the animal kingdom comes from things that look like you and I, 2 eyes, 4 limbs, so mammals and birds, and perhaps to a lesser extent, some bony fish and some corals actually, which is quite important and valuable information. And in terms of what we know about the plant kingdom, most of the information comes from showy, flowery plants with very little information being known for non-vascular plant species. Now, let me take you back to one of perhaps the most widely cited tenets of life history theory, and that is the fast law continuum, which was first put forward by Steve Stearns in the 70s. The fast law continuum emerged as a critique to RK theory. The main critique that RK had was that it was not really providing a mechanistic understanding of how species were evolving or were selected to either optimize R, intrinsic rate of growth, or K, the carrying capacity. So Steve Stern came up with some mathematical arguments and proposed that, in fact, there was a main axis of life history traits variation. That is, that if you collect demographic information for as many species as you will, and you organize them somewhere, somehow, you'll see that those photographs that I was talking about before will be correlated. And they will be correlated according to a single axis of variation, the fast law continuum. So species to the left are species that live fast and leave behind a beautiful cadaver. Those species invest a lot into reproduction, like C. elegans, but not much to survival. And as a consequence of that, they tend to reproduce a lot, but they don't live long. Species to the right-hand side correspond to strategies so-called slow, and they are pretty much embodied by species that allocate their limited resources mostly into their own maintenance, therefore securing survival, and not so much into the production of the next generation, aka reproduction. So those are the two extremes, and Steve Stern argued anywhere in between, you'll have strategies that pretty much capitalize on this key trade-off between reproduction and survival. Now, there have been attempts to try to tackle whether the fast law continuum does apply to plants as well. And I'm here showcasing some really exceptional work being done by Jonathan Silverton and Miguel Franco. This work really is what incepted the origin of Compadre back in the 90s, by the way. So Jonathan and Miguel came together and they explored in their first version of Compadre, which had about 100 species for which matrix population models had been published. They explored whether using the principle of elasticity, you could organize species, plant species, along this single axis of variation, the fast law continuum. Now, critically, one of the mathematical properties of the elasticities of population growth rates of a matrix population model with respect to the matrix elements. So each of these numbers correspond to rates of staying in the same stage or growing to the next stage or reproduction. The elasticities of population growth rates with respect to relative changes on those matrix elements add up to 1. And so this gave inspiration to Silverton and Franco to organize species of plants according to ternary plots, which they took inspiration from studies in geology, where soils, for instance, are classified according to percent of silt, clay, and sand. So what you have here is one of their key results, where they were able to organize species all the way from annual species like this one here. Yuri, can I ask you, can you see my cursor moving, my mouse moving?
Yes, it's perfectly visible.
Fantastic, thank you. So they were able to organize species from the bottom left, such as annual species, all the way to pretty long-lived trees. And the way in which this arc here is structured is that species on the bottom left have got population growth rates that are most reliant on reproduction. They don't really rely much on growth, and they don't really depend much on survival. So the elasticity that the process that controls most the population growth rate of annual species is reproduction. On the other hand, herbaceous perennial plants tend to rely quite a bit on growth, intermediate values of reproduction, and not so much on survival. And at the other extreme, you have got really long-lived species such as sequoias, whose main controllers, the main forces governing population growth rate, tend to be elements related to survival. So this was all great and fun, except that a couple of years following that publication, the study started to be quite criticized by other colleagues of mine. So the first criticism is that actually elasticities have got some nonlinearities that were not considered here. The second one is that because elasticities of population growth rates with respect to matrix elements by mathematical definition add up to 1, the fact that they add up to 1, it means that it's really difficult, if not impossible, to pinpoint trade-offs. Because if the elasticity of, say, reproduction increases, the elasticity of growth and survival must by definition decrease so that the summation adds up to 1. So that was criticized on that basis. And more recently, we also published a paper where we run some mathematical simulations. And what came out of those efforts is that this space here, which pretty much shapes the arc, is unpopulatable. We have not been able to find a matrix population model that is biologically inspired in a life cycle of a plant that would make sense, that would have intermediate values of elasticity to reproduction, relatively high to elasticity, and relatively low to growth. So from that perspective, a few years ago, some colleagues and I started to think, okay, so maybe we should just abandon the principle of elasticity to try to classify plant species and to try to ask whether the fast-low continuum really does apply to them or not. And one of the novelties is that rather than using elasticities, we decided to use life history traits. So again, going back to these photographs that I was telling you about. As I mentioned in a few slides ago, from any demographic model that I've presented to you today, you can apply age from stage decompositions, mostly methods developed by Hal Caswell, Steve Elner, and other folks, and derive key life history trait information such as generation time, that is the mean age of reproduction of individuals in the population. You can also, derive information about how quickly individuals in the population are senescent, the age at maturity, the number of offspring being produced per capita, the degree of heteroparity, that is how frequently do individuals in that study population reproduce throughout their lifespan, the net reproductive output, which is a measure of demographic success, the number of offspring produced cumulatively throughout the lifespan of the individual, the length of the reproductive window, for how many years does the average individual in a population remain actively reproductive, the rates of growth at the individual level, and also, importantly, because we're talking about species that have got the ability to decrease in size from one year to the next, the rate of shrinkage or negative growth. Now, pay attention to the colors that we're using here. So we're using white for generation time, we're using gray for life history traits that define longevity, we are using red for life history traits that dictate different moments of reproduction, and we're using blue to indicate changes in size. So the idea here is that we could put these traits in multivariate analysis and evaluate whether plants do really follow the predictions from the fast-low continuum put forward decades ago, which were not able to be tested decades ago because we didn't have the data, except now we do. In addition, because in the analysis that I'm going to present you today, we can and we did test for the role of phylogenetic proximity of different species in this multivariate analysis. We can start to decompose whether the demographies of different species are mostly affected by their ancestors through measures of phylogenetic inertia, or through the environments in which they have evolved. For instance, you have got two pine species here, bristlecone pine and the black pine. Obviously, they both belong to the same genus, but they actually live in very different kinds of ecosystems. One is found in an arid land, the other one is found in the Mediterranean habitats. The Pinus nigra, by the way, is a really successful invader in parts of New Zealand. So we can start to ask questions about not just are these life history traits aligned to a single axis of variation, but how does the phylogeny and how does the ecology of the species matter in that alignment? So what I'm presenting you here is a principal component analysis results showing you the first two axes, PC1 and PC2. Each dot corresponds to a natural population, so one species, and this analysis contains 415 species. They're color-coded according to the major ecoregion from which they were sampled. And together, these two axes explain less than 60% of the variation. One of the main key predictions of the fast law continuum is that a single axis should predict about 80%. And with not even with two, are we coming close to that 80%? So what this means is that the plant demographic data are a lot more dimensional than previous efforts that have focused only on animals, by which I mean really mammals and birds. On the first axis, which really can be thought of as a fast law continuum, you have got species to the left that grow fast, and species to the right that attain high generation times, high longevities, and low rates of senescence. And that axis absorbs about 34% of variation in the plant kingdom. And then the second axis is what we have come to call the reproductive strategies axis. In this case, it absorbs 21%. And it classifies species according to different moments of reproduction going up and the ability to decrease in size. So at the bottom, you have got species that don't reproduce much, don't reproduce frequently, and they don't have a really high R0, net reproductive value, and they do have a high ability to decrease in size from year to year. And at the top, you have got species that have got high per capita reproduction, long lengths of reproductive window. They're highly iteroporous, or if you are botanically inclined, polycarpic, and they have got high reproductive values. Following this research, I tested the same principles on more data. So this paper, a follow-up paper that was published a few years later, includes data from 650 plant species from compadre. And the same result came up even stronger in this case. So here you have growth, number 4 is growth, different moments of longevity and generation time going to the right, all 4 key moments of reproduction, perhaps you could argue 6, which is the per capita reproductive rates, are shared between both of them, and then 5, again, retrogressive growth or shrinkage. So even with more data, the picture becomes stronger. Well, okay, so we now know that at least for plants, we can classify their life history traits according to how fast they live and the modes of reproduction. What good is that for us in terms of predictive ability, right? A classification that doesn't help you predict emerging properties is as good as not having any classification. So it turns out that this key classification based on 2 different axes is quite useful. For once, it can help you predict population growth rates. Species with a high population growth rate at the top, species with a low population growth rate at the bottom. And it can also help you predict the ways in which natural populations can inherently deal with disturbances. I will not talk too much about it. Perhaps I'll do it for questions from the public later, but there are ways in which we can quantify the ability of a natural population to respond to disturbances. And that's through the usage of transient dynamics matrix. So what we can see here is that the degree of demographic resilience measured as per the damping ratio, which measures how quickly a population will go back to equilibrium after being perturbed, is maximized following 2 strategies. 1, species that live fast, and 2, species that have got a high ability to decrease in size. Perhaps when things get tough, either they live fast and they set seeds to recolonize the population, or they decrease in size in hopes that the next year it will be more advantageous and they will grow up again. As I mentioned before, this is a very active area of research in the group. We've published a few papers looking at these questions more in detail, and I'll be very happy to answer any follow-up questions from that. But in the meantime, allow me to expand onto the animal kingdom. I mentioned that when this type of analysis were done back in the 70s, in the 80s, folks, for the most part, had access to demographic data for mammals and for birds. Already in the 80s, in the late 80s, Jean-Michel Gaillard and Madame Oli showed that there was something interesting, something not quite adhering to the rule in regards to other traits that didn't quite correlate with the fast law continuum. Nonetheless, their analysis were constrained to mammals and birds. Kevin Healy, a postdoc that I co-supervised, led these efforts where we evaluated whether the fast law continuum is also the go-tell-all of animal demography. And the answer again is no. So on the x-axis, you have got the first PCA. This is also phylogenetically controlled, by the way. And this PCA absorbs 43% of that variation. Species to the left are fast and short-lived. Species to the right are slow and they are long-lived. But then importantly, we have got a second axis that absorbs 27% of variation. And just to make it clear, the previous analysis that I showed you had highly heteroporous plants at the top and highly monocarpic or semiporous plants at the bottom, species that reproduce just once, right? Thank you to reviewer number two. Reviewer number two, if you're in the audience, thank you very much for that. We had to revert the axis and therefore make this comparison a bit more confusing. So now we have got semiporous species at the top, like salmon, and humans as well. Humans are relatively semiporous compared to many other species of animals. And species towards the bottom, like crocodiles and turtles, highly heteroporous, are found, like I said before, at the bottom. So together, these two axes add up to 70% of variation. So it's not just one axis that will come close to 80% as predicted by life history theory. Knowing how fast or slow a creature is, and knowing its mode of reproduction also has got really important repercussions for our predictive ability for conservation biology. What I did here is I classified species, this is for plants in this case, according to these two traits. And then I interface the data with the IUCN Red List conservation status. Plant species here are classified all the way from invasive in blue, and those can be found mostly at the top and the top right, to data deficient. So those are species for which no conservation assessment has been carried out. So we don't know exactly whether they are endangered or not, all the way to an increasing risk of fashion, least concern, not vulnerable, vulnerable, endangered, and critically endangered. You can hopefully see that there is a gradient going from top left, be more likely to be invasive, to bottom left, be more likely to be critically endangered. And in fact, and if I may ask, please not to tweet this slide too actively, because this work is currently under manual revisions in a journal. We've been able to replicate this analysis going into high resolution for reptiles. This is work led by a postdoc in my group, Roberto Rodriguez, and what he was really interested in was not just to be able to do this decomposition of life history traits. In reptiles, the same answer comes out. There's an axis that explains about 40%, that's the fast law continuum. And there's another axis that explains about 23%, and that's the reproductive strategies axis. But also we wanted to evaluate interfacing with IUCN and other information that we have got access to, what regions of that two-dimensional area are more likely to go extinct based on different human threats. And what you can see is that it's mostly the areas in the periphery. Those strategies that are more unique are less represented overall in reptiles. Those are the species and the strategies that, if things continue to go the way they go, are more likely to go locally or globally extinct. So the part that emerges from this first part of the talk is that the fast law continuum really needs to be revisited, and the theory that underpins it as well. We should really think of the fast law continuum as a development to survival continuum with a novel added axis of reproductive variation, which is highly orthogonal from the development survival continuum. Now you may be thinking, wait a minute, when you presented me with data, I was not able to see any species populating this space or any species populating this space. And you're completely right. The first reason for the first lack of data here may have to do with the fact that those are incompatible strategies. So these are species that are neither allocated much to survival nor to reproduction. So of course they would have gone extinct a long time ago if they ever existed. The other area that is not very well represented has to do with species that will be quite long-lived, but they don't reproduce much. These are probably jewels of demography that have not yet been sampled across the tree of life and also around the globe. So if you know of some examples of demographic data that corresponds to this strategy, please let me know. I would love to test it and to see if we can actually expand this area over here. But common to these two axes, the fast law continuum and the reproductive strategies continuum, is that to place a species on it, we don't need any information about how the vital rates, the rates of survival, growth, and reproduction can vary through time with the environment. And I'm going to take a little kitty break because I've been talking for too long. All right. So obviously there's predictions about which life history strategy should be selected for fast living, slow living, highly reproductive, not so reproductive, depending on what kind of environment we are entertaining. But like I said before, all we need to position a species on that periodic table of life history strategies is a few years of data. We don't really need to explicitly accommodate environmental variation, which is it's quite important, right? Particularly in the light of what I'm presenting you here. Change is happening, and change is probably here to continue to happen. So which life history strategies will be affected the most by climate change? This is perhaps the most important question that population ecologists have to still address. What we know is that more extremes are already being recorded and more extremes are likely to continue to. Now, we also know that contrary to the existing theory to model populations in stochastic environments are not really truly stochastic. They don't really go into white nose transitions. So you can picture a population that can fluctuate between a good environmental year and a not so good environmental year, a bad year. In an IID environment, the probability that a population that say starts in a good year remains in a good year or goes back to a bad year, or a population that starts in a bad year remains in a bad year or moves on to a good year, it's all the same. It's purely stochastic, white noise. But what we know is already happening in terrestrial ecosystems is that there's an increase in positive autocorrelation, such that if the natural population that you start to study is already observed in a given state, it's more likely to remain in that state and then eventually transition and then remain in that new state. So the transition of change would look something like this. Bad year, bad, switch. Good, switch. Bad. So on land, we are already seeing an increase in the positive temporal autocorrelation of environmental stochasticity. Interestingly enough, underwater, the picture is quite different. We are seeing a proclivity towards a more negative autocorrelation. So previous PhD student of mine, Maria Panev, now an independent research fellow at the Estación Doñana in Seville, in Spain, led this effort where we simulated increases in the temporal autocorrelation in a positive fashion for plants and animals. So what I'm about to show you is an effort that includes both compadre, so that's plants, and comadre, that's animals. We first organize the species according to, as always, the fast law continuum and the reproductive strategies. And then we run sensitivities of the stochastic population growth rate of each population with respect to an increase in the positive autocorrelation of the environment, which is a mouthful, I understand. So allow me to simplify the language. We have calculated how vulnerable are these natural populations to the projections of climate change. And that's what we find. Again, from left to right, you have got increases in longevity, and from bottom to top, you have increases in reproductive window, increases in heteroparity. The thickness of each point, where each point is a natural population, it means an increase in vulnerability. So when this type of exercises have been done, assuming IID environments, it was the long-lived species that were more vulnerable. The novelty of this study is that when we actually take into account an increase in temporal autocorrelation, it's the short-lived species that are predicted to get to become more vulnerable, to become more sensitive to these increases in climate change. We've also been exploring these questions with marine data. Obviously, there's a huge gap in terms of high quality demographic data in the marine world. There's always limitations, such as the fact that we don't have gills. And so collecting demographic data, which is really hard work in Sirius, is quite limiting. However, we're led by a previous postdoc in my group, now Marie Curie in Spain, Paul Capdevila, have asked the question about whether life history traits and life history strategies really differ much between marine and terrestrial ecosystems. And the answer is yes. To such an extent that when compared to terrestrial species, the axis that matters most to marine species is not the fast node continuum. So what you can see here is hundreds and hundreds of plant and animal species characterized according to marine in blue and terrestrial in brown. What you can see here is that most of the variation of marine life history strategies is found along the second axis, not the first axis. We have got some ideas as to what that might be the case, mostly having to do with the viscosity of the medium in which these creatures have evolved, water, and the challenges of having predators around them from all different angles. Happy to discuss with you if you have got any questions. So there's nothing explicit about this environmental stochasticity. There's nothing explicit about environmental stochasticity in this framework, even though we can start to run simulations. The interesting thing is that recently, the topic of demographic buffering and lability has attracted quite a bit of attention from the discipline. We have got a few papers that have recently evaluated the ways in which different populations can buffer against this climate change, or just go with the flow, have vital rates that follow the changes in climate change. And the main take-home message of this part is that there seems to be a third axis that is highly orthogonal, for sure, to the heteroparity, similarity axis, also partly orthogonal to the fast low continuum. And that's what we now call the buffering liability axis. Tultia-Purkar developed a small noise approximation to the stochastic population growth rate of a population Landais. Without really wanting to get too much into the details, this equation can be simplified as it follows. The stochastic population growth rate, so the overall performance of a population in a stochastic environment, can be approximated as it follows. The mean deterministic population growth rate, that is the population growth rate of that population year after year, assuming determinism, minus something. In this case, is minus the temporal covariation of vital rates. But it's not just any vital rate. This S here is sensitivity, is the temporal covariation of the sensitivity of population growth rate with respect to vital rates. So the temporal covariation of the vital rates that matter the most, that control that population growth rate the most. From this small noise approximation, Cathy Fista back in the late 90s put forward the demographic buffering hypothesis. That is that being that it's pretty damn difficult to optimize this part of the approximation, the deterministic population growth rate, the part of this inequality where natural selection can operate more is on trying to constrain the covariation, the temporal covariation of those vital rates, which means this, that for any one population, the vital rates that have got a high sensitivity of population growth rate on the y-axis would be constrained in time the most. So this vital rate here for any one population would be highly sensitive of population growth rates and constrained in time. And this vital rate here will have a sensitivity to population growth rate relatively low, and therefore become a bit more invisible, shall we say, to population growth rate. But at the same time, we know that there's many other species, zooplankton, for instance, that show the opposite trend. And this is a term that Dave Coons has coined as demographically labile. These are species where, regardless of how high or low the sensitivity of population growth rates to survival, to growth, to reproduction may be, those vital rates will almost freely oscillate and will be fully at the disposal of that environment. In good years, the vital rates will go up. In bad years, the vital rates will go down. We now have got a really good understanding of the reaction norms that govern these relationships. We know that in species whose reaction norms between the environment and the vital rates follow a concave shape tend to be more demographically labile. We also know that species where that reaction norm is either linear or convex tend to show demographic buffering. But as I said before, this is an area that is quite active in the lab at the moment, and the evidence that we're starting to gather points towards the fact that there is indeed a third axis of variation. This is a paper that I published last year where I evaluated the different life history traits of plants in golden yellow, of non-human animals in blue, and of human animals in pink. And there was a third axis, which is this one here. You can also see a line with the second axis is the BL, buffering liability. What this is doing is quantifying the slope of that relationship for any species. If the slope is highly negative, the species is buffering. If the slope is highly positive, the species is labile. And we can see that even having information about how buffering or labile different human populations are can be a really interesting proxy to predict things like GDP. This is what is being shown on the second panel here. So the radius of each point corresponds to the population growth rate of each human population. These correspond to ISO 3 codes approved by the UN. So this is Austria, that's the Czech Republic, that's Lithuania, that's Finland, that's Malta, that's Greece. What you can see is that there is a trend whereby species that tend to be more buffering at the top have got a high GDP, for instance. So all these interesting properties are starting to emerge from really taking into consideration how natural populations vary or co-vary with environmental stochasticity. There's a paper that we published recently that talks more about these relationships. I invite you to take a look at it. And there's really a lot of predictive value in this framework. It takes three, rather than just counting how many electrons and how many layers of electrons, we need to count how fast or slow living creatures are. How does the schedule of reproduction, how does it get orchestrated throughout the lifespan of the organism, and how buffering or how labile different species are to the environment. On that, I like to be a bit shameless here. If you allow me to, I'd like to advertise a PhD position that recently opened in my lab. There's a QR code and more details. Please get in contact with me if you're interested in exploring this together. I'd like to thank Yuri for this very kind invitation, the initiative, which I think is a wonderful initiative, and also all of you for coming and joining me today. Thank you so much.
I would like to thank you, Rob, for the excellent talk. And with that, if anyone has a question, then what we do is please raise your hand in the participant list, and I will be calling on you. And if you have any questions, yes, please go ahead with that. Yes, Steve, please go ahead.
Hi, Rob. Very interesting talk.
Thank you.
This is touching on a lot of stuff that we're interested in right now, related to luck in life histories. So, but one of your slides worried me, and that was the one that had lambda on it, where you looked at it's a ways back, relative to the
Was it this one?
the reproductive strategies. Keep going. Unfortunately, yeah, that one.
Yep. Mm-hmm.
No, the one with lambda. You had it a moment ago.
This one.
There, that's it. Population growth rate lambda. Okay, and this is something we're wrestling with right now. Something with a lambda 0.5, negative population. It just shouldn't be there, right? Right, and a lambda of 1.5 can't go on for very long. So I wonder, is that population growth rate, or is that sampled in a good year versus sampled in a bad year?
So
and that mostly that your reproductive strategy axis is more about that than about anything else, and that might explain why that axis also maps onto the conservation status.
yeah, so we're starting to ask questions regarding intra-specific variation in the placement of species in this two-dimensional, now three-dimensional framework. We have got some preliminary evidence that the positioning of species along the second axis is more plastic than along the first axis. We also have got some preliminary evidence that that may indicate that the drivers of variation on the first axis and the second axis are different. Primarily, the first axis seems to be resource acquisition. The second axis tends to be aridity. But these relationships are quite weak at the moment. So we're very actively exploring that. As you know, Steve, and I'm really happy to hear that you're also interested in these questions. As you know, the status quo of animal implant demography is that there's not really a lot of spatial replication within the same species. So I feel a little bit reticent to tell you, it's definitely this or it's definitely that, because the reality is that we don't yet have a lot of species to run this analysis in a more robust way. Obviously, the databases, as you know, mostly because you're a partner in crime, continue to grow at a pretty fast pace. And the papers that are coming to the database these days are ones that tend to have more than 2 or 3 populations, which is fantastic. But when this paper was published, we didn't have that just yet, that degree of spatial variation. So I suspect you're right. And there's preliminary evidence that points that perhaps there's a lot of plasticity in the second axis. But even if there is plasticity on the second axis, I'd argue that that's really important to know. The framework is still very valuable, particularly if it still continues to correlate with things like responses to human threats, classification of the IUCN. In the IUCN, especially as some of these classifications lack quantitative assessments.
Okay, I'll make one final comment. I'd be interested to know how much of that plasticity, when you have the data, is spatial variation versus temporal variation. And I'll cede the floor with that. Absolutely.
I can tell you that more variation examined to date has to do with space than with time. But again, the same criticism, right? So we're so far we're a little bit too limited by spatial replication. We are actually doing better on the time departments than on the space departments.
Thank you so much. Giza, please go ahead.
My problem is something similar, that in a long term, average population growth rate should be around 0, which is arranged by population regulation, food level, for instance. And then so reproduction should compensate with mortality. And my understanding in this issue is that if if immortality would be an option, and then nobody wanted to spend energy on reproduction, but if there is a level of mortality which cannot be avoided by easily, then the reproduction should be high. And I think this is a kind of determining factor of reproductive strategy. Do you agree on this? Or
so I feel like allow me. Thank you for your questions. I feel like there's two questions there, so I'm gonna have to ask you for clarification on the second point. I come to the first point. Yeah, of course, everything. The log population growth rate should be zero. In fact, the log population growth rate should be negative because everything goes ultimately to extinction, right?
yes.
This framework, what I'm presenting here, is operating at shorter scale, ecological scales. Applications to conservation biology, no, they don't really care about where the populations are going to be in 50 years. They care about the short term, right? The second part is the one where I had a little bit of trouble following you. I think the noise was not very good. You were talking about a trade-off between mortality and reproduction. Do you mind elaborating, please?
No. Okay. Of course, there is some trade-off between investing in reproduction and investing in survival. But the question, what is the factor which determines the outcome of this trade-off? And I suggesting that there is a minimal kind of minimal level of mortality which cannot be easily avoided, then sufficient amount of energy should be spent on mortality or reproduction, otherwise the population will die out. Or phrasing differently, if the unavoidable mortality is high, then the population should decrease to a level when the food supply allow larger reproduction for the equilibrium. And then so it is very important to take into account this feedback, population regulation feedbacks, and and it is the part of the very essence of the situation.
Yeah,
you cannot. Okay.
So I think I now understand what your point was, but please correct if I'm not. Still, the sound from your mic is not coming very clearly. I apologize for that. So two answers to that. The data that I showed you before, we have got heaps and heaps of data, right? So we've gone through a really careful selection of species into this analysis. We require that at least 5 years of demographic data be available for us to be able to decompose these life history traits. Within those 5 years, implicit to them, there's density regulation already. Okay, it is true that more elaborate work needs to be done in the field of demography. Funny enough, because demography, I mean, density is the bread and butter of it all, right? And many times it's not being taken into account explicitly. The second point that I might want to make about it is that you're talking about the trade-off between reproduction and mortality. Where? We don't see that at the interspecific level, right? When we're thinking about investment, so trade-offs are underpinned by two components of the economics of the organism: investment and resource allocation. And of course, I'm referring you back to classical work by De Jong and others. Identifying trade-offs is really difficult. The picture that emerges from this type of analysis, whether we do that for the entire plant kingdom, the entire animal kingdom, or subsections, is that the moments of reproduction tend to be largely independent from the strong trade-off that we do find, which is if you develop fast, you die young. And if you develop slow, you live long. Independent from that trade-off, There's how you structure your reproductive strategy. When do you start to reproduce? How long do you reproduce? How often do you reproduce? In each reproductive bout, how much do you reproduce? All of that seems to be largely independent from that initial investment, initial trade-off between development and survival. Hopefully I've made justice to your question, Gisa. I apologize if I didn't quite understand it. Happy to continue chatting though.
Thank you so much. Brian, please go ahead.
Yeah, I enjoyed the talk and enjoy this whole body of work. It's really like how you pull the data together and finding these generalities. I had a question. So, these principal component analyses of life histories, it matters a lot what you've controlled for, right? The original Stearns paper, I think that we were talking about, found 80% in the fast slow. I think, if I recall correctly, didn't control for body size. So it wasn't just that it was birds and mammals, it was also that it didn't control for body size. And Jessica and Coco had this paper that showed if you do body size or phylogeny, you can get very different trade-off axes depending on what you're controlling for. And if I recall correctly, most of your papers control for both phylogeny and body size, and that's state of the art. I'm not critiquing that, but I just wonder if you could comment on how much depends on those corrections, and in particular, as we start to take this back into the applied context, for example, response to climate change or conservation vulnerability, whether body size, phylogeny controlled corrected life history is really the most relevant factor, or if the raw uncorrected body size included is more relevant at that point.
Thank you so much for that question, Brian. That question is really brilliant. The corrections so even if I might have said the word correction, I actually hate the word correction in this context, because I don't think saying that you've corrected by phylogeny means that you actually have done something to get rid of the nuance that phylogeny is. But I think that both phylogeny and biomass bring a lot to understanding how demographies have evolved and how they respond to environmental variation and human threats. Brian Inquest once put it to me as a challenge to be able to decompose how much body mass matters compared to other factors. And it matters a lot. In fact, when we do a regression boosted tree analysis on life history traits in plants, the first thing that gets selected for is body mass. You can criticize, of course, that within the plant kingdom, our estimate, our proxy to body mass is less than optimal. We are decades away from knowing truly how heavy a sequoia is. We are decades away from knowing how heavy Artemisia transdentata is. It's a proxy based on above ground height. What happens below ground remains a big mystery to us. And we also know that root to shoot ratio are quite different in tropical ecosystems compared to desert ecosystems, right? In fact, when you look at herb in a desert, you're really looking at something really tall, most of which happens to be below ground. Having said that, though, we have evaluated that, as you know, in the analysis with and without these considerations of body mass, the signal remained the same. We've also done the analysis with body mass in it, just to kind of like, sorry, my headphones just died. Can you hear me okay? Fabulous. Sorry, my headphones just died on me.
Yes.
So we've also done the analysis, the multivariate analysis, where we've considered phylogenies explicitly and now put biomass in the analysis. And body mass is really correlated with generation time, both in animals and in plants. So in terms of conservation managers, body mass tells you a lot about the positioning on the first axis, doesn't tell you anything about the second axis or the third axis that we're proposing, the demographic path frame versus liability axis.
Yes, thank you. Any other questions, comments? So yeah, just one thing that I was wondering during the talk, but then you answered the question as for whether these matrices have sort of implicit density dependence in them, because the way they're measured, they cannot be factored out. But presumably it's not explicit, right? It's amalgamated into the numbers that go into the matrix. Is that right?
That is absolutely correct, Yuri. So it's quite a half-glass perspective on demography when you look at the way in which some of these demographic tools have been parameterized, and you realize that a lot of it is not explicitly density dependent. I'm looking at Steve and he's nodding.
I think it's fine. There's nothing wrong with it. It's
well,
just
there's nothing wrong with it. I would like to make a plea to folks working in demography in year 2022 to start to make things a bit more explicit, because we know density matters, right? Having said that, though, there's a few models that are now entering the database that do that explicitly, and that's great. IPMs, Padrino is doing things a lot more by the book. So a lot of IPMs that enter our database are density dependent, and that's fantastic. In regards to the matrix population models, what we do is, rather than just collecting one and do an analysis with that, because that might happen in a good year or a bad year, right? So that may not be representative. It doesn't take into consideration, therefore, either implicitly or explicitly intergenerational trade-offs, nor density dependence. So that's where we usually constrain our analysis to those data sets for which we have got longer runs.
Exactly. So thanks for clarifying that. And I was wondering if that at least goes some ways to explaining the strange figure in air quotes that Steve pointed out. That and you also mentioned that in your view, temporal variability matters less than spatial variability. But and that might be so, but I'm just trying to put the puzzle pieces together from what you were saying. You also said that it's quite important that we have at least for some organisms, the positive autocorrelation. So then if the time of observation is a 5-year window, say, then is it possible that we catch that population within a window? And therefore, yes, temporal variability is not that important over a, whatever, 15-year long time scale, but over the time scale of this observation, maybe it's a bit more important. Is that a possibility?
It is absolutely possible, and this is why the macroecological approach cannot really act independently, right? So what we need is a combination of really carefully elaborated experimental analysis on one species, replicated through time, following that long term, coupled up with a macroecological approach, which is what I presented here mostly. We've got a set of experiments both in lab and in the field where we are testing these questions more under control of experimental manipulations, right? Macroecology is great for patterns, not so much for mechanisms.
Yes, thanks.
It's entirely, going back to your question, Judy, it's entirely plausible that a few, if not quite a few of the species that we have portrayed in those analyses happen to be on a good run for 5 years or on a bad run for 5 years, right? It's the hope here, and that's the ethos of the macroecological approach, is that the wealth of data will iron out those differences.
Okay, yeah, absolutely. Thanks so much, Rob. By the way, I have to mention one more thing. I really hope that somewhere you've already written down the phrase "from humans to bacteria, from lions to dandelions." That's basically poetry. Thank you very much for that.
Thank you. It is part of the intro of a book that I wrote 2 years ago on demographic method.
Excellent. Thanks. In any case, are there any more questions for Rob?
There is a question in the chat, maybe.
There was a bunch. Yes. An argument about lambdas, a paper. Yeah. Maybe Rob, you can say something about that. I remember there was a question about a paper that shows.
Oh, let me see which one. Do I take it from the top? Or which question should I go? Okay, so correct that study.
By the way, just while you're looking to reflect to what Geza is saying, yes, of course, it's trivial on the long run. The question is how long it takes for it to converge.
So
and
it's not, so the question to the paper that I think I apologize, I don't know who Andras Hulai. The question that I think Andras is asking, the first question, so do I remember correctly, Rob, that I have seen a figure of yours showing that with NPMs, generally the longer the length of the study has been, the closer to lambda. Yeah, so that's partly true, but the variance and the convergence to lambda equal 1 depends quite a lot on the strategy. So annuals, it takes way longer than, for instance, trees. Mammals take shorter than it takes, for instance, for things like insects. So generation time really is a better predictor of the convergence to lambda equal 1, more so that's the replication of the study.
Thanks. This reminds me, by the way, I had another question, sort of a trivial little thing. What are the units in the compadre systems?
So these are discrete time models, right? Most of them are annual models.
Okay, so they're
So
yearly.
folks go to the field every year.
Okay.
Most of them. It really, the time unit is commensurate to how fast living the creature is. So obviously, if you want to follow the demography of C. elegans and you go there to your petri dish every year, you can miss everything. But flip side of the coin, if you go to Sequoia National Park and you go there every year, you're not going to see anything.
Yeah.
Change is too slow. So there are matrix population models that are parameterized every 5 years, every 10 years, mostly for the giants of the tree of life, sequoias and other trees. And that's fine. There's things that we can do to butt convert those models into annual.
So that would have been a question, that is there an easy way to know what are the time units for any one given organism and to automatically do the unit conversion?
It's all archived in the database, and we've developed quite a bit of our functionality to run the analysis that I presented to you today are actually fully open access in our GitHub.
Fantastic.
And it allows you to take subsets of data according to your search criteria and then automatically rerun like search traits and all these time conversions and things like that. Mostly we started doing this mostly from a selfish perspective because we thought, this is really hard work, so let's just make sure that things are done well in a way that is more automatic. But then we realized great value to the audience, so let's make it open access as well.
In that case, may I please ask you to put can you type a link into the chat?
Yep.
Thanks so much.
So in fact, let me see, I will have to, so I'm gonna put, hold on a second. This is, so there's two packages that we developed recently. One is called rcompadre. This is the GitHub to rcompadre.
Oh, is it on CRAN?
Yes.
Okay.
And there's a methods in ecology and evolution publication from last year that introduces both packages. And
thanks so much.
Let me just, so rcompadre methods. And this is the paper.
Mm-hmm. Okay.
In case you're interested in it.
Fantastic. Thanks so much. Yeah, are there any final questions for Rob? If not, then thank you so much again for the great talk, and thank you everyone for coming. And I believe we will be continuing with the seminar in 2 weeks, but maybe somebody has the schedule up.
It's Melissa Guzman on, I think something meta communities. We haven't got a title quite yet.
Okay, yes. Thank you for the information. So hope to see you there. And until then, thank you again, and see you next time. Bye-bye. Bye, everyone.